collaborative project between my team at the Michigan Kidney Translational Medicine Center and some of our friends at the University of Washington, where we benchmarked optimal cell type label transfer settings for spatial transcriptomics. And I also want to acknowledge the improved type 2 diabetes study, the renal air study, the crocodile study, Neptune, and the George M. O'Brien Kidney National Resource Center for their contributions to this project. What really excites me about spatial transcriptomics is that unlike single cell and single nucleus RNA-seq, which dissociate the cells from the tissue, spatial transcriptomic keeps the cells within their native tissue context. And one of the top assays right now is Xenium. And Xenium is an image-based spatial transcriptomic technology that achieves subcellular resolution. Unfortunately, that high resolution comes at a cost of not being able to measure all of the genes within the transcriptome. But you can take single cell data, which has cell types labels defined by a transcriptome-wide signal, and you can map that transcriptome-wide signal onto a reduced spatial gene space to get your cell type labels onto your spatial transcriptomics data. And what's cool about spatial transcriptomics data is that then, since the cells are still in their native context, you can verify the plausibility of cell type assignment by looking at how it appears on the tissue itself. And so that sort of sets up my benchmarking study. And so we have 3 kidney cohorts. We have the Improve Type 2D, the Renal Air, and the Crocodile, which is youth onset type 2 diabetics. And then we have Precise, which is kidney nephrectomies. And we also have Neptune, which are nephrotic syndrome patients. We're taking those 3 kidney cohorts. We are applying the cell types from those cohorts to 4 Xenium kidney nephrectomy samples. And we are doing that with 2 algorithms, Seurat and Tanagram. So the Improved Type 2D renal air crocodile data is all single cell, and the Precise nephrectomy data is also all measured through single cell. But what's really interesting is that 4 of the single cell samples are the same individuals that were assayed with Xenium. So we can look at both the whole cohort as a reference data set and individual matched data sets for the label transfer. And finally, Neptune is the largest cohort represented within these data. It's mostly single nuc RNA-seq, but there is some single cell. So we're going to analyze that data as a whole, and also with the single cell and single nuc data split to look at how the technologies differ. So to sort of review this slide again, we're going to end up with 5 single cell and single nuc kidney data sets. We're going to take the cell types from those, and we're going to apply a label transfer with Seurat and Tanagram, and we're going to map those cell types onto our Precise samples, which are from the 377 Xenium gene panel. And the first thing we wanted to look at is how well are the counts from the genes correlated across the technologies. And so the plots here, each dot is one of the 377 genes that is assessed within Xenium. On the x-axis are the Xenium gene expression counts, and on the y-axis are the single cell or single nuc gene expression counts. And what you can see is across cohorts and data sets, the correlation between the genes is high across technologies. And now I want to go in, and our first evaluation of which algorithm did the best is to look at the label transfer confidence scores. And that is along the x-axis here. And it's apparent right away that Seurat produces higher confidence predicted cell type labels. If you look and you are comparing these darkly colored dots with the pastel dots, especially in the Seurat section, you can see that merging into lower resolution cell type labels improves the scores. And so by doing that, I mean if the single cell or single nuc data has perhaps 2 T cell populations, to sort of just merge that into 1 T cell population improves the label transfer confidence scores. And now just to zoom in on Seurat, which from the label confidence, does seem like it's doing a little bit better. I also noticed that the single cell reference sets are actually scoring higher than the single nuke reference sets. So the single cell are the blue dots and the single nuke are the orange dots. And what's interesting about that is if you remember in Neptune, the single nuke is actually represented by more than 100 people. So you might expect, based on a power calculation, that it would perform a bit better. Another thing that we notice is that if you're comparing the squares with the dots within the precise, is that we are getting higher confidence label transfer when we use the complete cohort versus just a matched individual reference set for the label transfer. And that sort of leads into saying that it looks like the highest scores are from the largest data set with the best phenotypic match, which is the precise data set using the whole cohort. And then the second thing is, those are just probabilities, and we really want to be able to look at these cell types on the tissue to verify if they are plausible and sort of making sense. And so what I'm showing you here are some of the transferred labels, and I'm showing you the highest scoring tanagram samples so that we can have tanagram looking as best as it can. And what we're looking at in the orange are parietal epithelial cells. And those are expected to ring the glomerular structures here, which are these circles, sort of in a halo. And so you can see that Surratt is correctly identifying cells that are plausible to be these parietal epithelia or PEC cells. Whereas tanagram is capturing some of those same cells, but it's also nonspecific because you can see a lot of cells scattered in non-plausible locations across the tissue. And so that just shows that the Surratt labels both have a higher confidence score and look more plausible on the tissue than tanagram for these kidney samples. And now I wanted to show you a little bit about different immune cell resolutions based on the reference data set that you choose. So on the left here, well, in both sections, I'm showing you from the same sample we saw on the last slide. And here I'm showing you an immune infiltration. And so we're looking at just a cluster of immune cells. And on the left are just the unsupervised clusters from out of the box xenium. And that is mostly giving us natural killer T cells. But when you apply a label transfer from the precise single cell matched individual data, in orange, you will see that there are some B cells in that area. And so you're getting a higher resolution immune cell type than you would with just unsupervised clustering. And just to zoom in really quickly and show you that there are B cell markers in the area. So I do think that these are correctly identified B cells. Next, I wanted to sort of look at how the matched individual versus the complete cohort on the right compare on the tissue. And what I'm seeing in the complete cohort is that we're getting more orange cells. So we're actually able to recover more B cells with the complete cohort. And I believe that's because the B cells are quite rare. They're only about 1% of the reference data cell population. And so by having the complete cohort, we have more power to correctly identify B cells. And finally, I wanted to look at how the difference technology underlying the reference data alters immune cell types. And so what we're seeing on the left is that with single nuke data, you don't get a high resolution of immune cell types. And so it's really only identifying some macrophage and monocytes and some T cells. But on the right, you get a whole mosaic of different types of T cells. You can separate the monocytes, you can detect B cells. And so selecting your reference data set, you'll want to definitely be very thoughtful about the labels contained in your reference data as those will apply directly to your spatial data. So thanks for listening. It looks like Surratt label transfer is accurately inferring kidney cell types. And that the best data set to use for reference is the largest phenotypically matched single cell or single nuke, and that you'll want to be really thoughtful about the reference data technology as that will alter the resolution of certain cell types. And what's really cool is that since we can view these transferred cell labels in their native environment, spatial transcriptomics is giving us a ground truth to benchmark the plausibility of these cell labels and to be able to evaluate algorithm performance. And I want to thank my co-authors again, and a special thanks to Neptune, the George M. O'Brien Kidney Center, and Renal Air Crocodile Improved Type 2D for their financial support and or providing the cohort level kidney data for this study. Thanks again.